Hello everyone, so today we have the lecture first part of the lab, and it is about the forensic DNA fingerprinting. So what we are going to talk about is, first of all, that we are going to use the kit, special kit, with all needed materials and substances supplied with this kit. So first of all, to understand the lab, we'll need to talk about the DNA structure, then the restriction analysis. After that, we're going to talk about the agarose gel electrophoresis, how the molecular weight is determined according to the samples that we obtain in the end of the gel electrophoresis. Of course, the practical part is the simulation of DNA fingerprinting, and also we will need to talk about the plasmid mapping. So what is the fingerprinting of DNA? So we identify some sample of DNA somewhere, and we want to determine the owner of this DNA. So let's say that it is again the crime scene. So we found the DNA samples like follicles of hair or skin parts, etc. And we also have some suspects. And from these suspects we can obtain the DNA samples and then compare them to the crime scene. So what we do, we use the restriction enzymes to identify the fragmenting of DNA samples, and if they are, so let's say this is the crime scene, and here we would have the suspect 1, 2, 3, 4, and 5. So let's say that suspect 3 has actually the same pattern of fragments on the gel. Okay, so it will tell you that the suspect 3 has the same fragmentation of DNA as the person from the crime scene. And based on these analyses, we can firmly say that, okay, so the third suspect is actually the person who was on this crime scene. So what else we could do? We could do the relatedness of humans, and let's say that some people, they want to identify the father of the child, so identification of the paternity. Same goes for animal relatedness. Also the analysis of DNA samples from distant ages. So let's say that we found bones and we could collect some DNA from it, and we want to say if it is human or not. Of course, by doing this restriction enzyme analysis, we could identify the species for this bone. Also, we could perform the research on disease-causing organisms, and in this case, usually we are talking about the viruses and bacteria. What else? We could identify the source of food. So let's say that some person wants to know if they actually eat beef. So they send this food for this identification, and then according to the animal relatedness, they obtain the results saying, okay, so this is beef or not. Of course, human remains work the same as anthropology studies, so you identify if the remains of the organisms are actually belonging to humans. And the monitoring of transplants, of course, now we have better methods to monitor the transplants, but before we could obtain them, we were using the RFLP method very widely in transplantation. Okay, so during this lab, today's lab, we are going to talk about the restriction digest of the DNA. Then we are going to talk about the DNA fingerprinting and RFLP analysis. After that, we are touching the electrophoresis, and it is going to involve the agarose gel. So agarose gel is usually performed with the samples of nucleic acids. So RNA and DNA, I usually go with the agarose gels. Finally, we are going to perform the analysis and the interpretation of the results. Okay, so first of all, let's talk about the composition of nucleic acids, and in this case the deoxyribonucleic acids. So as you all know, we have base pairs, and the pairing happens according to the hydrogen bonds formed between the nucleotides. The pairing happens in the way A to T, and we have 2 hydrogen bonds, and G to C with 3 hydrogen bonds. Of course, because we want to show it in a simpler way, we usually write A to T and G to C without showing the 2 hydrogen bonds and 3 hydrogen bonds here. So here is the formula and the spatial formula. So here you can see the phosphate, the deoxyribose, and the nucleotide itself. And we have cytosine and guanine here. So they are forming these 3 bonds between the hydrogen and oxygen here, hydrogen and nitrogen here, and hydrogen and oxygen here. And if we're talking about the thymine and adenine, they form only 2. So here you can see the H being attracted to oxygen, and hydrogen from the second hydrogen bond is attracted to the nitrogen here. We have already talked about the composition of DNA in chromosomes on the Genes in a Bottle laboratory. So again, the chromosomes are the DNA molecules wrapped around the histones. And here you can see these proteins, histones, and the DNA, if you unwind it and make it the tether-like structure, so it's only the secondary conformation, it will look like this tether. DNA will be wrapped around these histogen bits, okay? Then they will form the chromatids, and the chromatids will form the chromosome. Okay, so what are the restriction enzymes or restrictases? They are coming from the bacteria, and why it evolved in the bacteria? Because they don't have, since they are the unicellular organisms, they don't have immunity to the invaders. So they produce the defense system themselves. So every single bacterium produces the defense system themselves. So in this case, we are in need to protect ourselves against the viruses. So here we have bacteriophage, and this bacteriophage injects the DNA. So through this injection, the viral DNA gets into the cell, but if the bacterium has already started the production of restriction enzymes or endonucleases, this viral DNA will be cut in different places, and in this way the bacterium is saved from the invasion. For now, we have over 3,000 known enzymes working as the restrictases. So here you can see the endonuclease, and endo means that if you have the DNA molecule or RNA molecule, endo means that the cut will happen somewhere in the middle, not in the middle, but away from the edges, okay? So we have edge here and here. So anywhere between these edges is called endo, inside. If you have the digestion from the edge. So let's say that cut happens here, then here, and it goes on, which means that the enzyme comes and bites the pieces of the DNA. It will be called exonuclease. Okay, so we have the special sites of recognition, and these sites of recognitions are specifically recognized by the restrictases. And if we take the ECOR-RI, so this is the one that we're going to use, it will identify the site which goes GAA-TTC. And if you look at the complementary strand, you will see that it is actually the palindromic sequence, so it goes GAA-TTC. So in this case we will have the so-called palindrome, and it is repeated palindrome, and the enzyme will cut it not with the blunt ends. We will have the sticky ends here, so we will have overhangs on one of the strands. If the 5' is shorter than the 3' end of this fragment, then it's called 5' overhang. So for ECOR-RI, we are producing the 5' overhangs. Okay, so these sites of recognitions, they're called restriction sites, okay? And as I said in the previous part, we are producing the 5' overhangs. So if the 5' end is shorter than the 3' end, it's called 5' overhang. In contrast to the ECOR-RI, we have the PST1, and PST1 produces the 3' overhangs, and the sequence is CTG-CAG. So if you create the palindromes, it will be CTG-CAG. So it goes the same in both directions. Okay, so the source of these enzymes, of course, the ECOR-RI is Escherichia coli, PST1 is the Providencia stewarti, and we are going to use these two enzymes in our lab. So what else you should know about these restrictases analysis? And it is about the buffer, the restriction buffer, and this buffer creates the special conditions for these restrictases to work. And for that we will need the sodium chloride, which creates the ionic strengths, and the ionic strengths along with the pH produces the special conditions required for the best conformation of the enzymes. And the magnesium in this case, so the ion of magnesium, it is the cofactor of the enzyme. So without magnesium, the enzyme will not get the shape that is required for the restriction function. So let's say that without magnesium it looks like that, but if we add the ion of magnesium, it will change the conformation, and now it could actually cut the DNA at the restriction sites. Okay, so what else you should know about it, about the restriction itself or digestion, that is that we need the special temperature. In the case of ECOR-RI and PST1 restrictases, we need 37 degrees of Celsius. Why? Because it is optimal body temperature of the Escherichia coli, they live in the colon, so that's why they are having the optimal temperature range, which is very close to 37 degrees. If we have too high temperature, the enzyme might undergo the denaturation, or in other words, it's killed, so because it cannot perform the functions. If it's not high enough, so the temperature is low, we have the low activation of the enzyme, so the functions will be very slow, and the restriction will not happen in the all possible restriction sites. So of course in this case we will require much longer digestion time. Okay, now let's talk about the restriction fragment length polymorphism, or shortly RFLP. So what is polymorphism? Morphos means shape, poly means different or a lot of. So in this case we're talking about a lot of fragments. So let's say that we have the gene in one person, and allele 1 has the sequence that could be cut by PST1. Okay. On the allele 2 we have the sequence that has point mutation, and it's not critical, and this point mutation does not allow the function of the PST1. So instead of AT we have CG here, and of course the PST1 will not cut the DNA here. So what happens on the restriction site of the EcoRI, let's say that we have the same sequences that will be allowing the action of EcoRI. So what happens here, as the result of this cut, we will have short, then we will have long one, and again we will have short one. So this one will be the restriction site of PST1, and this is EcoRI. Okay, so the allele 1 produced 3 different pieces. The allele 2 will produce one long and one short. So this is the EcoRI restriction site, and of course we have 2 fragments and 3 fragments here. So if we are doing the gel electrophoresis here, we will have different number of fragments obtained with the same restrictases. As you can see here, so for the second allele we will have one long part, and of course because it's long it did not travel much from the well, so this is well, and the distance it traveled is related to its size. So what happens to the second part? It is very short and it travels a lot. Okay, so it get very far from the well. So if we take the allele 1 fragments, we will have the same part on the right end, and of course it's seen here. And also we will have the parts, 2 parts that are cut, and they should be smaller than the long part of the allele 2, and of course we see it here. So if you imagine the bond here, it will be the same bond as this long piece, but because we have the cut somewhere here on the allele 1, it will create 2 smaller fragments, and this is the polymorphism, or different fragmentation of the genes. The marker lane here shows the kind of standard of the cuts And of course, if you take the DNA content in the well at the beginning of electrophoresis, we will have different sizes of the DNA here, and we will be able to compare them to our samples, DNA samples cut by the restrictases. So here we have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 different size fragments of DNA. Okay, this is the end of today's lecture, and for the next week's lab we will have a short lecture related to the plasmid mapping and the gel electrophoresis.